In this video, we're going to work on the ALEKS problem called Understanding the Glycosidic Links in Common Polysaccharides. There are two versions of this problem. I'm going to show you one version, and I'm also going to talk about how you would complete the different version of the problem. So in this version, you're going to be given usually 4 monosaccharides connected together to make a polysaccharide, and you're going to be asked a couple of questions about that structure. So I've got the questions over here. In addition to the 2 questions, you're asked to highlight the glycosidic bonds in the molecule. So there is a highlighting tool. It's going to be underneath, it's going to be located down here underneath this drawing box. So you're going to click on that highlighting tool and highlight the glycosidic bonds. The glycosidic bonds are the bonds that connect the monosaccharides together. And my first instinct when I was solving this problem was to highlight these bonds right here. Unfortunately, that's not exactly what ALEKS wants. ALEKS only wants you to do a partial highlighting of the bond. It just wants you to highlight the first half of the bond, which is kind of hard to describe which half is the first half. But if you're looking at a bond that runs horizontally, you're just going to highlight the left half of the bond. And if you're looking at a bond that runs vertically, you're just going to highlight the top half of the bond. Don't highlight the whole entire thing. It just marks it wrong and doesn't give you very good feedback. All right, so once you get the bonds highlighted, then you are asked to write the symbol for each type of glycosidic bond that is in the molecule using the standard notation for a glycosidic bond. You can see you've got your Greek letters over here as well as a little arrow tool. This particular polysaccharide has 2 different types of glycosidic bonds. These 2 are identical, so that's one type, and then this is a different type, and we're going to practice coming up with the symbol for each of these bonds. So to come up with the numbers for the symbol of a glycosidic bond, we're going to be using the numbers of each individual monosaccharide carbon skeleton. And this numbering comes from starting at the right-hand point of each ring, so starting right here, and then going clockwise around the ring. The 2 carbon atoms that are connected by the glycosidic bond are going to give us the 2 numbers in the symbol for the glycosidic bond, and we list them in numerical order, smallest one first going to the larger one, and they go into parentheses. Then for the letter outside the front of the parentheses, we're going to look at the carbons, look at the 2 carbons of that glycosidic bond, and we're going to ask ourselves, or we're going to determine whether either of these carbon atoms were anomeric before these monosaccharides came together to make a polysaccharide. An anomeric carbon is a carbon that's on the right-hand point of its individual ring. So this carbon right here, because it's on the right-hand point of its ring, this carbon is anomeric. The OH group that was at one point on this particular carbon atom is sticking up, which means that this is the beta anomer of this particular monosaccharide. This carbon atom is not an anomeric carbon. It is not the carbon atom on the right-hand point. The anomeric carbon is carbon number 1, in this case, the one on the right-hand point. So this is not an anomeric carbon of this particular glycosidic bond. This is the only anomeric carbon, and it has the beta orientation because the oxygen is sticking up, not down. And we're going to write that symbol, beta, in front of the parentheses. As I said, both of these glycosidic bonds, these 2 glycosidic bonds are identical, so they are both beta 1,4. This glycosidic bond is different, so it's going to get a different symbol for it. We're going to number the individual monosaccharide, or the individual ring. So we can see that this glycosidic bond connects carbons 1 and 6. So we're going to put that in parentheses. The bond goes from carbon 1 of one ring to carbon 6 of the other ring. For the symbol out front, we have to determine whether either of these carbons were anomeric. An anomeric carbon is the carbon on the right-hand point of the ring. So this was an anomeric carbon, and its oxygen is sticking down, which means that it was an alpha carbon. This is not an anomeric carbon. It is not located on the right-hand point of its ring. So this is an alpha 1,6 glycosidic bond, and that we would put in this space right here. Now, the last question is asking us whether or not that polysaccharide could be any one of these 4 different types of polymers. Now, I have looked at a ton of these different problems, and none of them involve DNA or RNA, so you can just rule that out immediately, and that leaves you with 4 different possibilities. Cellulose, all of these contain glucose. Cellulose is a polymer that contains beta-1,4 glycosidic bonds. And we have beta-1,4 glycosidic bonds in our polysaccharide. However, cellulose contains only beta-1,4 glycosidic bonds. It doesn't have anything else in it at all. So this is not cellulose. It's similar, but it is not cellulose. Amylose, glycogen, and amylopectin all contain alpha. I'm going to figure out how to write this. All of these contain alpha-1,4 glycosidic bonds. They will all have that. The amylopectin and glycogen, so these two, I'm trying to figure out the best way to organize it, will also contain alpha-1,6 glycosidic bonds. So if you have something that has only beta-1,4 bonds, you know that it is cellulose. If you have something that contains only alpha-1,4 bonds, you know that it's amylose. If it contains both alpha-1,4 and alpha-1,6, it could be either glycogen or amylopectin. You're going to want to check both of those. Now, in this problem, this particular one that was given to me, I have 2 different types of glycosidic bonds that are not consistent with any of these 4 substances. So I think that this is a trick question, and I think that I am not supposed to check any of these boxes at all because the polysaccharide that I have here does not match up perfectly with any one of these 4 things. So this, I believe, is a trick version of this problem. Now, the other version of the problem, the ALEKS is going to start you off with 3 random structures of molecules. One of them is going to be kind of a monosaccharide. The other 2 are just going to be completely random looking things, totally irrelevant. You're going to want to delete them right away. It is going to give you a structure of something that is kind of a monosaccharide. It may be glucose, it may not be glucose, it may be something random, and it's going to ask you to draw a polysaccharide, a specific polysaccharide. So it's going to ask you to draw one of these 4 polysaccharides. So you're going to take that glucose monosaccharide that's been provided to you, you're going to delete the other 2 things that it gave you. You're going to first check and make sure that this glucose monosaccharide is actually glucose, because I've seen some versions of the problem where ALEKS gives you a not accurate version of glucose. Once you've got the molecule in the form of actual glucose, then you are going to connect at least 4 monosaccharides to match up with whichever polysaccharide ALEKS is asking you to draw. So for example, if it's asking you to draw cellulose, you want to connect 4 glucose monosaccharides in a beta-1,4 linkage. If it's asking you to draw amylose, you're going to connect 4 glucose molecules in an alpha-1,4 linkage. And if you're asked to draw either glycogen or amylopectin, those 2 structures are going to end up looking the same.